Welcome to The Explainer. Today, we're diving into a science that's not just stuck in labs. It's in your kitchen, it's in our medicine, and it's changing our world. We're talking about genetic engineering.
But before we get into all the high-tech stuff, let's start with a really simple question. Have you ever made curd, yogurt, or even just baked a loaf of bread at home? Well, if you said yes, then congratulations. You're already a biotechnologist. Turns out, using living things to make useful stuff is something humans have been doing for, well, thousands of years.
So how did we make that massive leap from baking bread to actually editing genes? That's the journey we're going to take today. We'll start with the big idea, then look at the two core principles that made it all possible. We'll open up the genetic toolkit, check out the assembly manual for building new DNA, and finally get into why all of this really matters.
Okay, first things first. To really get a handle on the power of this science, we've got to be on the same page about what biotechnology even means.
So, the European Federation of Biotechnology gives us this official definition, and you'll notice it's pretty broad, right? That's because the field is just massive. It covers everything from the microbes in your yogurt to the most cutting-edge tech in today's gene editing labs.
And this right here breaks down the key difference perfectly. Think of it this way. Traditional biotech uses the whole organism, like all the yeast in a batch of dough. But modern biotechnology zooms way in. It gets incredibly precise because it works directly with DNA, the source code of life itself.
So, what is it that allows for that incredible precision we just mentioned? Well, it really all boils down to two core principles that completely kick-started the revolution in modern biotechnology.
The first one is genetic engineering. This is the main event. The idea that you can deliberately rewrite an organism's genetic code to change how it looks or functions, its phenotype. It's kind of like editing the blueprint of a building to change it from a house into a skyscraper.
Now, the second piece of the puzzle is bioprocess engineering. You know, if genetic engineering is designing the recipe, this is building the massive high-tech factory. It's all about creating these perfectly clean, controlled environments to grow your newly designed organism and make tons of useful products, like vaccines or life-saving antibiotics.
Okay, so if you're actually going to edit the code of life, you need more than just a good idea, right? You need a very specific set of tools. So, let's open up the genetic toolkit.
First up, you need molecular scissors. Seriously, that's what we call them. They're these amazing enzymes that can make incredibly precise cuts in a DNA strand, right at a specific spot. This lets scientists snip out the one single gene they're interested in.
Next, if you have scissors, you're going to need some glue. And in our toolkit, this is an enzyme called DNA ligase. Its job is pretty simple. It pastes that gene we just cut out into a new piece of DNA, sticking all the fragments together.
And finally, you need a way to get your newly made DNA into a living cell. For that, we use what's basically a delivery truck, or a vector. One of the most common ones is a plasmid, a little circle of DNA from bacteria that's perfect for carrying a new gene into a host. We've got our scissors, we've got our glue, and we've got our delivery truck. Now for the really fun part. Let's look at the assembly manual and see how scientists actually use these tools to build something totally new: recombinant DNA.
This whole process was first figured out way back in 1972 by two scientists, Stanley Cohen and Herbert Boyer, in a truly groundbreaking experiment. And as you can see here, they used every single tool in our kit. They took their molecular scissors, cut out a gene for antibiotic resistance, used their molecular glue to paste it into a plasmid, and then used that delivery truck to get it into E. coli bacteria. The result? A brand new bacterium that was now resistant to antibiotics. It was absolutely historic.
And that amazing experiment basically established the three-step playbook we still use today. First, find and cut out the gene you want. Second, get that gene into your host organism. And third, and this is crucial, make sure the host actually accepts the new DNA and can pass it on when it reproduces.
But here's a really interesting problem. You can't just drop a new piece of DNA into a cell and expect everything to work. Just getting it inside the cell isn't enough to make it a permanent change. So, why not?
Well, the reason is that any piece of DNA needs a special signal, a specific sequence called an origin of replication, or ORI for short. Think of it like the start button for the cell's copy machine. If that new gene isn't attached to an ORI, the cell's machinery just floats right by it, and the new DNA never gets copied.
This brings us to our final and maybe most important section. We have the tools, we have the manual, but why is this modern approach such a revolution?
Let's see how it just completely blows past what we could do with the old methods.
You know, traditional breeding is kind of a genetic lottery. You cross two different plants hoping for the best, but you usually drag a whole bunch of unwanted genes along for the ride. Genetic engineering, on the other hand, is like performing surgery. It lets you insert only the specific gene you want with no extra baggage.
And that's really the heart of it. This modern approach gives us a level of control that was just unimaginable before. It's a total game changer, swapping guesswork for absolute surgical precision. So if you remember just one thing from our time today, let it be this word, precision. The entire incredible power of modern biotechnology comes from the ability to make specific, targeted, and intentional changes to the code of life itself.
And that's where we are today. We now have the tools to edit the source code of life. That toolkit is in our hands. Which leaves us with the biggest question of all. Now that we can do this, where do we even begin?